Welcome back to my course Aspects of Biochemical Engineering. In the last lecture, I tried to give the information on different bioproducts. As you know, bioproducts of there are different varieties. Different classifications we have. We have low value, high volume products, medium value, medium volume products, and high value and low volume products. Now, on the basis of cost, this product may be classified. Now, today I will be starting one very interesting topic, which is very essential for mass and energy balance of any kind of biochemical processes. The topic that I am going to discuss, that is stoichiometry of biochemical processes. Now, what do you mean by stoichiometry? Stoichiometry means that interrelationship different components present in the reaction mixture, and also it gives the information that how much that heat evolve that we have in the biochemical system. As you know that in the biochemical process, most of the biochemical processes, they are exothermic in nature. So, exothermic means they are heat liberating. So, most of the biochemical processes are usually operated at ambient temperature and atmospheric pressure. So, it is close to 30-35 degrees centigrade. Now, particularly since we are in the tropical country, during summer the temperature rises to as high as 40-45 degrees centigrade. So, naturally, since we have heat evolution in this particular bioreactor or biochemical processes, so we should have more cooling arrangement. Question comes, how much cooling arrangement is required? That can be determined if you find out how much is evolved in the biochemical process. So, stoichiometry not only give you the information of intermediate molecular relationship of the different component present in the reaction mixture, but also heat evolve in this process. And also it gives another information which is very important in the biochemical process, that is the validity of the experimental result. Because we carry out some kind of experiments in the laboratory, and naturally the results that we get, that how to evaluate that whether the experimental data that I am reporting, that is we are reporting, that is right or wrong. So, these are the three different information we can get from the stoichiometry of bioprocess. Now, here if you look at, so what I told, it is like this, that that the stoichiometry of the biochemical process give the following information: study of quantitative relationship between among the reactant used and the product forms of a chemical reaction, or mutual relationship and internal limitation within the biochemical system. That means, what I want to point out, how much of the substrate molecule gives how much of product, that theoretically that you can find out from the stoichiometry. Then another thing I told you, the validity of experimental results, and third one is the heat evolve, particularly in the aerobic fermentation process. Now, what do you mean by law of conservation of mass? Conservation of mass, if you look at here, that is rate of substrate inflow and rate of substrate outflow, and this is passing through a system like this. Now, that means a law of conservation of mass, that nothing can be destroyed, nothing can be killed. The overall balance of the process that should be remaining same. Now, here if you look at the mass in through the system boundaries and minus mass out through the system boundaries, and plus mass generated within the system minus mass consumed within the system is equal to mass accumulation within the system. This is called conservation of mass. Ultimately that should remain same. Now, that when you do the stoichiometry of the bioprocess, that we shall have to do some elemental analysis, and through this elemental analysis we can find out how much of substrate molecule reacts with how much of A, B, C, D, how much of A reacts with how much of B reacts to give how much of C and how much of D. So, this is you have some kind of elemental analysis we have. Now, if you look at this elemental balance, the material balance, if you look at material balance of the biological reaction can be easily written within the composition of substrate, product and cellular mass are known, because so that in the biological system, because since we are talking about the biosystem, what is happening in the biosystem, you are putting your substrate, then you are getting the product. Now, when you get the products, your products may be of different, either in the form of cell mass, or maybe if it is cell mass, mostly you get the cell mass. If you are other than cell mass, so you get cell mass as well as the product. Now, usually the electron proton balance are required in addition to elemental balance to determine the stoichiometric coefficient of the. This I shall discuss in details how you can find out the stoichiometric coefficient of the biochemical processes. This is very interesting. Today, determination of the composition of the cellular composition is the major problem. So, what we do usually, we assume that kind of cellular composition of the biomass. This is CH 1.8, O 1.05 and N 0.2. It is found within 5% limitations. It gives the empirical that formula like this of the cell mass. So, another very important thing is 1 mole of biological material is defined as 1 gram atom of carbon, that is C H alpha, O beta and N delta. Now, let me explain that, because suppose we are talking about glucose C6 H12 O6, this is, we call it glucose. Now, when you write this glucose in the form of this 1 gram atom of carbon, how you can write? This is CH2O6. So, 1 gram atom carbon is of glucose is like this. This is 1 gram atom glucose when you use it as a substrate. Am I right? So, this is similarly it is applicable. Suppose any compounds, when you determine the empirical formula, we can easily convert in the terms of 1 gram atom of carbon. This we can easily convert in terms of that. That is not very difficult. Now, here is the let me show you the macroscopic biomass balance of the microbial system concerning the biomass production, and another product can be written in the original form. So, that when you write any kind of stoichiometry of the bioprocess, it is essential that we should have some preliminary information on the system. As for example, I want to point out, suppose we want to produce some kind of Baker's cyst. Now, in the Baker's cyst fermentation process, we are targeting to produce the biomass only. We are not targeting maybe carbon dioxide is a kind of a byproduct of the system. As you know, the carbon dioxide that produced in the Baker's cyst fermentation process, or in case of ethanol fermentation processes, we have more or less in the purified form, and it is used for the production of dry ice. So, so commonly that how we can write this equation. If you look at, this is the empirical formula of substrate. This is CHMOL. This is the substrate. Now, when you talk about biomass, we required some kind of nitrogen source. So, usually we put nitrogen in the form of ammonia. So, we can consider a mole of ammonia that is required. Then if it is the aerobic system, so aerobic system we required molecular oxygen. So, we can write BO2, this is molecular. So, and in what we get the products like biomass. This is the empirical formula of biomass, this is the empirical formula of product. So, YC is the YC moles of gram atom of biomass and YP gram atom of product, that is equal to, now it is kind of carbon balance. You can see that 1 mole of carbon is converted to YC moles of biomass and YP moles of product. So, what will be the carbon dioxide? 1 minus YC minus YP, this CO2 and plus CH2. So, but suppose we want to produce some kind of biomass. So, we can ignore the other than the product formation. Here the product formation that we have, that we can ignore. But if we want to produce some kind of product, as for example ethanol fermentation process or citric acid fermentation process or penicillin fermentation process, then we can ignore this biomass formation. But if it is the anaerobic system, we know anaerobic system, that amount of cell mass produced is drastically reduced as compared to that of aerobic system. So, if we ignore that, your relationship will be simple. One thing is that this oxygen will not be required, the nitrogen also mostly will not be required, because if you do not consider biomass, the product formation. So, your carbon source is mostly converted to your product, if product does not contain any nitrogen and also carbon dioxide and water. This is but in case of aerobic fermentation process, when you consider the cell mass, then you can ignore the product formation. This you can ignore, and you can write, this is the substrate, this is the ammonia, this is the carbon dioxide, this is oxygen, molecular oxygen, this is biomass, carbon dioxide and water. So, this kind of, suppose I can give you another example. Suppose in the anaerobic digestion process, the organic biomass, organic mass is converted to methane and carbon dioxide. Now, when organic mass converted to methane and carbon dioxide, what we considered, since it is the anaerobic process, we should not consider any kind of oxygen, because it is oxygen is harmful for the system. And also the anaerobic system, we do not produce a significant amount of biomass. So, nitrogen source is also we do not consider. That what we considered, since this organic residue is a polymeric material, so we required water molecule for the hydrolysis of this initial hydrolysis breakdown of the polymeric molecule. Then when you get the monomer, it is converted to methane and carbon dioxide. So, what will be the equation you have? You have substrate plus water equal to methane plus carbon dioxide, that you will get in the system, maybe with a hydrogen ion, that excess hydrogen ion might be present there. This is how you can write the balance equation. Now, let me discuss about the degree of reduction. What do you mean by degree of reduction? Degree of reduction is basically, it is the free electron present in any kind of reactant or product. Now, question come, how you calculate the free electrons, because we considered that carbon, if you look at carbon, what is carbon? It has 4 electrons in the outermost orbit. So, you can take 4 more electrons, we consider C4 plus. Now, in case of hydrogen, we can consider it is 1 plus. Am I right? Now, in case of this oxygen, it usually take 2 electrons. So, it is minus. So, the 4 electrons can go out, 1 electrons can go out. So, it is plus, and this, since it can take 2 electrons, we can add nitrogen. What we can write, that in the outermost orbit it can take 3 electrons. So, it is 3 minus. So, you know that we can write like this. Now, if you now if you this is I told you this is the formula of glucose and 1 gram atom. The how if you do the electron balance, free electron, this is 4 plus 2 minus 2, because 2 into 1, because this is basically 2 into 1. So, it is coming around 4. So, 4 is the free electrons. So, degree of reduction basically it deals with that how much free electron present in the particular substrate molecule. Now, suppose if for ammonia, carbon dioxide and water, we consider the degree of reduction is 0, that is the free electron is 0. So, when we write, so we can suppose we write a empirical equation like substrate plus oxygen give the kind of product, or biomass, you can say biomass. So, plus carbon dioxide. So, that we can write the balance equation, how much that free electron present in the substrate, how much free electron present in the oxygen molecules, how much present in the biomass, how much this will be 0. So, maybe if some product is there, then you find out that how much free electron. So, we can write a balance equation that particular, that I shall show you how you can do that. Now, here I have let us go here. This is, you see, the degree of reduction is defined as the number of free electrons in that quantity of the material contains 1 gram atom of carbon. Now, biomass, that if you look at the biomass, that this is biomass, where this is the formula of biomass, CHPONNQ. Now, if you look at this, that how you do this balance, therefore 1P, 1 hydrogen, and 2N, that is oxygen, and 3Q is the nitrogen. So, this is how you can write this nitrogen, the free electron balance for with respect to biomass. So, it is we express as the gamma B. Now, product, similarly we can write the balance in case of product. If you look at the product, that the product also you have this formula, that product has so what will the product, that is this is the product. So, this is 4 plus R minus 2S minus 2T. This is how we can write the, and similarly substrate we can find out this. Now, where the number of free electron taken 4 for 1 atom of carbon and 1 for hydrogen, minus 2 for oxygen and minus 3 for nitrogen. This we shall have to remember, and no free electrons of the metabolized end products, that end products we have like water, carbon dioxide, ammonia, while oxygen O2 accept 4 electrons. Now, here I have given some typical examples of elemental composition of different biomass. I can this is the different like Escherichia coli. This is the empirical formula we have. Then Klebsiella aerogenes, like Klebsiella aerogens. So, different type of and that respective degree of freedom. So, I ask all of you, you can do the exercise and try to find out whether you are getting this value or not in case of different types. So, this is the different, and that is why on the basis of that we select that average composition empirical formula of the biomass is ACH 1.79, O 0.5 and N 0.2. Now, in case of Escherichia coli, that if you look at, this is the formula that we have, just now we have found out, and what will be the free electrons? This is 4, then 1.777 into 1, then 2 into 0.49 and 3 into 0.0, so it is coming about 4.07. So, I hope it is clear to you. Now, we have done some degree of reduction calculation with respect to some substrate molecule like alkanes, alcohol, carbohydrate and organic acid. This has come like that. You can practice. Now, oxygen demand, how you can calculate, because this is very important. So, so I have already shown that our common equation is this. This is substrate plus ammonia plus oxygen. It gives biomass product, carbon dioxide and now, if we do the balance, this is with respect to substrate. This is the degree of reduction of substrate. Then we know that one O2 can accept 4 electrons. So, I can write B moles of O2, that is B into 0.4 minus 4, and then YC is the YC moles of YC gram atom of biomass. So, this is YC gamma B and YP gamma P. So, I can write that oxygen demand, this is B is the oxygen demand, we can calculate like this. Now, again if we divide both sides by gamma S, we will get that like this. The now 4B by gamma S is considered as the fraction. This considers the fraction available electron transferred from substrate to oxygen, and this is similarly this is we consider the fraction available electron transferred to substrate to biomass. This is biomass and this is product. So, again this is fraction of electron that is transferred to substrate to this is biomass. So, that so this is oxygen, this is substrate, this is product. This is how electrons are transferring. So, we have 3 different portions here. Now, this if you look at previous one, that this particular this portion, if you look at this portion, this is called energetic growth yield, and this is like this. This is eta, the YC gamma P by y gamma S we consider as this is called energetic growth yield. This is how we can write it. Now, if you go through that second portion, that here we can find out this portion. This portion we call energetic product yield. If you look at this is called energetic product yield. So, energetic product yield YP gamma P by gamma S, that we can do that like this. Now, another way we can differentiate, we can differentiate this biomass product and substrate, that is on the basis of what weight fraction of carbon. Weight fraction of carbon, because let me explain that. Suppose that I was talking about glucose. Glucose is CH2, H2O. So, gamma fraction, that weight fraction of carbon. So, what is the atomic weight of 12, and what is the molecular weight? 12 plus 2 plus 16. So, this ratio is the weight fraction of carbon. So, that you can do it very easily. So, this is similarly in case of biomass, I can do that. In case of product, we can do that, we can do in case of substrate. Now, biomass yield, that another very interesting thing, that is, let me explain that. Now, biomass yield is very interesting. Y x by s is what is equal to mass of cells per mass of substrate consumed. Now, this is equal to, actually this is equal to eta sigma s gamma s, then we have gamma b and sigma b. B stands for biomass. Now, question ka how this equation has come. Now, this we can easily find out that like this, that if you look at the eta we have already seen that eta equal to y c into gamma b by gamma s. Am I right? So, here if you put y c gamma b by gamma s multiply by you have gamma s sigma s and gamma b and sigma b. So, the gamma b and gamma b will cancel and this will cancel. So, what will basically we have, this is we have equal to y c into sigma s by sigma b. And if you what is this? This is the atomic weight of carbon by molecular weight or gamma atom of substrate. This is the atomic weight of carbon by gamma atom of biomass. Now, when you do that, your biomass will go there and you multiply by that, you will find, you will come across this equation, you will come across. So, you can easily find out how it has come. So, this is how it has come. This is now, similarly, we can calculate that other thing also, that y x by s is like this, and then y p by s is like this, epsilon p gamma s gamma say, sigma s gamma s sigma b gamma b. So, p this with respect to product we can find out. So, and gamma y p by s is equal to p minus p 0 s 0 minus s. So, we can easily calculate that. So, you know that another thing I want to point out, in case of biochemical system, thermodynamics yield coefficient, that coefficient efficiency of the process plays very important role. Now, in thermodynamic efficiency, in case of aerobic system, it varies from 0.5 to 0.6, and in case of anaerobic system 0.7. Now, this will help us to find out the validity. I told you the stoichiometry help us to find out the validity of the experimental results. So, this will help us. And another thing I told you that stoichiometry also determined how much heat evolved in the biochemical process. The Q equal to 4 Q0 into B, and the Q0 is equal to 133 kilojoule per equivalent of free electron transferred into B. So, this unit is kg per gamma atom substrate consumed. Now, if you divide this you can convert into, if you divide by molecular weight of substrate, then it will be heat evolved per gram of substrate consumed. Now, if you divide by yield coefficient, you can easily find out how much heat is evolved per gram of the cell mass production. So, this is this is the what I want to tell about the preliminary part of stoichiometry. And here we get the information that how we can develop the intermolecular relationship with respect to elemental balance, with respect to electron and proton balance, and then we find out that validity of the experiment result, because on the basis of this thermodynamic yield coefficient I just show you in next couple of lectures, and also we can find out the amount of heat revolved in this process. Thank you very much.